Welcome so much at Medical Sciences Made Easy by Naphtali Muhumuza. In the previous videos, we have seen the anatomy and physiology of the mouth. We have covered the histology of the GIT. And now we want to see the anatomy and physiology of the stomach in this session. Please always remember to subscribe, like the video, and share your comments so that we can keep developing this channel. It is our channel. We are learning medical science. Let's get started. So, in this, we want to look at the anatomy first of the stomach. Then we shall see the physiology. And we see the stomach is J-shaped. It is J-shaped. And it is the one that connects the esophagus, with the duodenum. That is the stomach. And this stomach, it plays a big, important role, whereby the functions of the stomach, one of them is storage. It acts as a reservoir that after taking food, rushing like some of us eat very fast, it is first stored in the stomach for 1 hour. And that 1 hour it is stored within the fundus. This stomach does the role of killing microbes that enter with the food with help of the hydrochloric acid. This stomach does the role of mixing food. It mixes food bolus with gastric juice. And this one also produces gastric juice. And this gastric juice is the one that contains HCl and intrinsic factor of Castle. And this intrinsic factor of Castle is the one that enables absorption of vitamin B12. So whenever you take food with vitamin B12, it binds with intrinsic factor of Castle produced by the parietal cells and is the one that enables absorption of vitamin B12 within the intestines. And we see still there is production of, it has G cells, and these G cells produce gastrin hormone. And this gastrin hormone is the one that triggers the production of gastric juice. So those are some of the functions of the stomach. And this stomach, apart from being J-shaped, apart from connecting the esophagus and duodenum, it is distensible. It is the part of the GIT that can extend. It is flexible. So the more the food you eat, the more it extends. So it is flexible and it is distensible. And we're going to see that this stomach, it is made up of 4 regions. It has 4 regions. And these 4 regions include the first one, the opening of the stomach. It opens via what we call the cardiac sphincter. The cardiac sphincter is the one that regulates entry of the food bolus from the esophagus into the stomach. That is the cardiac sphincter. And the first region of the stomach is the cardia. We have the cardia. Then the second region is the fundus. Fundus is always found above the cardia, where the esophagus joins the stomach. This is the first one, the second one. The third region, which is the largest, is the body. So body covers 75% of the stomach. So that is the body. Then the fourth is the pylorus. Pylorus region, and this pylorus region is divided into 2, whereby we have the pylorus antrum and the pylorus canal. And this pylorus canal ends up onto what we call pyloric sphincter. And this is the one that regulates gastric emptying. That pyloric sphincter, which is found here at the end of the pylorus canal, is the one that regulates gastric emptying into the duodenum. So these are the four regions of the stomach. And these four regions of the stomach, which is the cardia, fundus, body, and the pylorus, they are the one that we are going to see how each plays an important role. So we have said the cardia is the first portion. This one is the first portion. And it is the one that connects it. It is the one that leads to the fundus, whereby fundus plays a big role of storage. It is the one that stores food bolus. In fundus, whenever we swallow food and it reaches in the stomach via cardiac sphincter, they are stored by the stomach for at least 1 hour. Food is reserved or is stored for 1 hour before it is mixed with gastric juice. And it is this time of storage in the fundus where salivary amylase completes its action of hydrolyzing starch to maltose and dextrins. That is during the first hour after swallow. That in the fundus, the food here, there is no production of gastric juice. Hydrochloric acid is not yet secreted. And we know that this salivary amylase cannot work in acidic conditions. So for the first hour, it completes its action of hydrolyzing starch. Then after 1 hour, we see the mixing wave coming in, triggering the production of gastric juice, which mixes with the food bolus to form what we call food chai. So we see the mixing is done in the body. So the body is the one that does the mixing, whereby there is churning action of the oblique, the inner oblique, the middle circular, and the outer longitudinal, mixing the food bolus together with the gastric juice. And in this stomach, we shall see also production of gastric lipase. And this gastric lipase is the one that will start its action. And still before gastric lipase, there is also another one produced in the mouth called lipase. And lipase does not act in the mouth. It is produced in the saliva, but it acts in the stomach because it is activated by hydrochloric acid. For it to act, it works in slightly in the pH between 6 there. So when there is small production of hydrochloric acid, then lipase also hydrolyzes short chain tags. That lipase acts on the short chain tags, short chain triglycerides, and it breaks them to diacylglycerol plus free fatty acid. That is the action of lipase in the fundus major, as we shall see. Then, so we have seen the major thing in the body is the churning action, the mixing waves, mixing the gastric juice together with the food bolus. And here the end result in this body is what we call acidic food chyle, what we can call chyme. That is what results into the body. Then the antrum, we shall go and continue to see that the antrum, for it, its major work is that it contains G cells which produce gastrin hormone. And this gastrin hormone, I've not told you that in the body of the stomach, there is cells, enteroendocrines, they are cells, and one of the cells in the body, we shall see them, is the parietal. Then we shall see also chief cells. And these parietal cells are the one that release gastric juice. Gastric juice is produced from the parietal cells. Now, actually, in the parietal cells, gastric juice is a collective action. In the parietal, we produce hydrochloric acid and intrinsic factor. Whereas in the chief cells, we produce enzymes like pepsinogen, enzymes like gastric lipase, and prorenin in children who are still breastfeeding. And these hormones are produced as zymogens, that is inactive, 4. Whereby, so when we are talking about pyloric antrum, is that this gastrin hormone produced from the pylorus antrum is the one that goes and triggers these cells to release their secretions in the stomach. So they are secreted in the presence of gastrin hormone. But we shall see other cells in this place, a cell known as enterochromaffin cells, and we shall also see enterochromaffin-like cells, whereby enterochromaffin cells, for them they release serotonin, whereas enterochromaffin-like, they produce histamine. So these are the cells which are there. And this, you were told that histamine and serotonin are produced in the mast cells. So within the mucosa, the muscularis mucosa, we see there is some lymphatic vessels and some cells supplied there. That is the mast cells which have this enterochromaffin, which are part of the pylorus. So they are the one releasing serotonin and histamine. So we shall see that this gastrin, it is the one that is stimulating these two cells to release their secretions, and this gastrin is potentiated by histamine. So histamine is producing synergistic effect. It is the one, so gastrin hormone, together with acetylcholine, produced from the nervous system, they are the one that trigger production of these secretions, hydrochloric acid, intrinsic factor, these enzymes, in the presence of histamine as a synergistic effect. So histamine is like a Mularo booster, and its receptors in the stomach, its receptors majorly are expressed as H2, not H1. And we shall see drugs which target this, like ranitidine, H2 inhibitors, or what we call H2 blockers, like ranitidine, cimetidine, which are given in people with ulcers. But before we continue to look at the functions of each, we want to see how parietal cells can produce hydrochloric acid. We have seen the regions, now we want to enter into the physiology to see how does these cells produce their secretions which form part of the gastric juice triggered by gastrin hormone. So if I have a cell here, this is my parietal cell. This is my parietal cell. And this is the blood vessel. And this cell has receptors. It has receptors. It has receptors. And this is the basolateral membrane. Basolateral membrane, whereby the whole this space is the interstitium. This is the blood vessel. And then this is the parietal cell. This is the parietal cell. And this side is the stomacal rumen. The rumen of the stomach. So what happens is that we have some transporter here. That within the rumen we have always potassium ions. And these potassium ions that on binding, when gastrin hormone, the presence thought, that is thought of food, presence of food, smell, taste, is going to trigger what we call the nervous system to release acetylcholine. And this acetylcholine comes and binds on its receptor on the parietal cell. Then the presence of food in the stomach, the stretching also causes production of gastrin hormone from the G cells of the pylorus antrum. And this also has its receptor. So we see it also coming to bind. This is gastric hormone. So when it binds, we see these potassium ions in the rumen being transported inside the parietal cell, and as they move in the parietal cells, we know there is water combining with carbon dioxide. And when it combines, it forms weak carbonic acid. And this weak carbonic acid formed, it occurs in the presence of carbonic anhydrase enzyme. Carbonic anhydrase enzyme catalyzes the binding or the combination of water and carbon dioxide to form weak carbonic acid. And because it is weak, it dissociates to give hydrogen ions plus bicarbonate. And we shall see this hydrogen that will increase in hydrogen ions within the parietal cells will come and is released in the rumen of the stomach using, so we see the hydrogen ions being released. And this requires energy in form of ATP, whereby ATP is hydrolyzed to ADP. And this pump, which enables the proton to enter into the rumen of the stomach, is what we call potassium stroke hydrogen ATPase pump. ATPase pump. This is the one that enables release of hydrogen ions from the parietal cells to the rumen. How the water and the carbon dioxide join in the presence of carbonic anhydrase enzyme to form weak carbonic acid, which dissociates to give bicarbonate and hydrogen ions. And these hydrogen ions, they are brought inside the rumen in exchange with the potassium. That as potassium is entering into the cell, by active transport, it exchanges with the proton, what we call the hydrogen ion. Then the bicarbonate formed, for it exchanges. In the blood, we always have chloride ions, and these chloride ions come and enter into the parietal to exchange with the negative charge of the bicarbonate, which goes into the blood circulation. We know that the charge must be balancing. So why does chloride shift into the cell? Is to equalize the negative charge lost by the move of the bicarbonate. And this bicarbonate, when it reaches in the blood, is the one that causes what we call alkaline tide. Then this chloride, which shifts to equalize the negative charge, continues and is brought into the rumen. And when it comes in the rumen, we see it combining with, comes and marries together with the hydrogen ion, whereby they combine to form hydrochloric acid. And that is how it is formed in the stomach. So parietal cells do not produce hydrochloric acid. They produce hydrogen ion separately, and they produce also chloride separately. So we see them combining together when they reach within the stomach room. And that is how they are released. After the binding of gastrin hormone together with acetylcholine. But we said what potentiates or what gives energy, these two compounds, that is gastrin hormone and acetylcholine, is histamine, produced by enterochromaffin-like cells within the pylorus antrum. Then we saw also serotonin hormone being produced by the enterochromaffin cells, which is a strong vasoconstrictor. That is how we can. So this is how hydrochloric acid is produced. So in summary, as we conclude the physiology, we are going to look at different cells found in the mucosa lining of the stomach and their functions. Whereby we have seen their cells. So we are going to have cells, then what they secrete, and the function. And these cells are found within the mucosa. And the mucosa of the stomach which is lined by the simple columnar epithelia. We see other cells like in the stomach, we have cells like mucus cells, surface mucus cells, surface mucus cells, and mucus neck cells. Mucus neck cells. This is cell number 1. And these mucus cells, they are the one responsible for secreting, let me make it even one, the function. They are responsible for secreting mucus. They're the one that secretes mucus in the stomach. And this mucus is the one that protects the stomach walls from being destroyed by the hydrochloric acid. So these mucus lines, they seal the lining of the stomach so that it is not destroyed by the hydrochloric acid. Then we see these are the surface mucus cells and the mucus neck cells. And these cells, they secrete their mucus via gastric pits. We find also in the epithelium we have what we call gastric pits. That's why they secrete them. Then cell number 2, we saw parietal cell. And this parietal cell, we said it produces hydrochloric acid, but it produces it in the form of proton and a chloride. And then another thing, it produces also intrinsic factor. Intrinsic factor, and we said that this intrinsic factor is the one that enables absorption of vitamin B12 in the ileum. That's why you see people with chronic ulcers, or what we call chronic peptic ulcer disease, they can suffer from what we call pernicious anemia. This pernicious anemia, I can write it here. This is the spelling. Pernicious anemia is whereby there is reduced HB level, and when you do a film comment, you find the cells are bigger. They are megaloblastic. Why? Because there is no absorption of vitamin B12. It leads to what we call pernicious anemia. And in physiology labs, this test we can confirm pernicious anemia by doing Schilling's test, whereby we give a radiolabeled vitamin B12 both via the oral and the IV. Then we see the rate of their absorption. That is Schilling's test to rule out pernicious anemia. And before I leave the parietal cells, we have seen doctors treating people with the PUD by giving them what we call proton pump inhibitors, what we call PPIs. And these PPIs, their mechanism of action is that they act by blocking the potassium stroke proton ATPase pump. That whenever I take esomeprazole, I take omeprazole, lansoprazole, and pantoprazole, these guys, they come and sit on this pump, and they block the release of hydrogen ions into the stomach. And you know the problem is there is much production of the acid. So whenever I block production of the proton, the hydrochloric acid cannot be formed. So that is why we treat those people with the PUD sometimes using proton pump inhibitors, which we have omeprazole, the lansoprazole, the pantoprazole, all those ones, which we end with those. So they are all proton pump inhibitors which act by blocking the potassium stroke proton ATPase pump. Then from here we can see the third cell, and the third cell is the chief cell. The stomach still have the chief cells. These chief cells produce enzymes like pepsinogen. They produce prorenin. Renin, this is capital N. It is double N. It is different from renin from the kidneys. Then there is another known as gastric lipase. And these first two are produced in active form, and what activates them is hydrochloric acid. Hydrochloric acid activates these enzymes, and once activated, we see pepsin hydrolyzing the long chain the polypeptide chain of proteins to small peptide chains. That is the work of pepsin. Then prorenin, we said it is found in the children. You know, children are taking milk in liquid form. So what renin does when it is activated, it coagulates or it corrodes this milk into a solid form that can easily be digested, found in children. Then there is also gastric lipase, and this gastric lipase works also on short chain triglycerides. That when I have gastric lipase, it works on the short chain triglycerides. Only 10%, its action is 10%, and it breaks them down into MAG, that is monoacyl glycerol, plus free fatty acid in the presence of water. That is the use of gastric lipase. Then renin, coagulating milk proteins in children, pepsin, converting long chain polypeptide into small peptide bond. Then we saw also the function of intrinsic factor, but we never talked about the function of hydrochloric acid. The function of hydrochloric acid, for it does a big role in providing acidic pH. Provides acidic pH. It kills the microbes that enter with the food, like bacteria. It also activates enzymes. Inactive enzyme activates enzymes. And as if that is not enough, it also denatures the proteins. It denatures the proteins. Or the best word I can use, it unfolds proteins. Remember, proteins like a quaternary and a tertiary structure, these proteins are much folded. So whenever you take a protein, it reaches in the stomach, hydrochloric acid denatures by unfolding to expose the peptide bond. So that when pepsin comes, it can easily start breaking the peptide bonds. So another role of hydrochloric acid is to unfold proteins to make them to expose the peptide bond for action of the enzyme. So those are the four functions: acidic, providing acidic pH, killing the microbes, activating the pepsin and prorenin, then unfolding the proteins to expose peptide bonds for easy hydrolysis. Then another secretion, another cell, number 4, we saw G cells produced from the pylorus antrum, and their secretion is gastrin hormone. And please, these cells are very much examinable. So we can ask in objectives, we can ask in section B or section C, telling us which that outline cells in the stomach and the secretions they do secrete, and their functions. Or we can ask in objectives that which of the following cell secretes gastrin hormone, which of the following cells secrete hydrochloric acid. So it is very important to master the function of each as a medical student. So we said that this gastrin hormone, once it is produced, it binds on the parietal cells and triggers production of gastric juice. The secretion is produced by the chief cells, and the parietal cells are the one that forms the gastric juice. Gastric juice, all secretions from parietal cells and chief cells form the gastric juice, and all of them are triggered by gastrin hormone together with acetylcholine potentiated by histamine. Then another cells we saw is the enterochromaffin cells, and we said these guys, for them, they produce serotonin, and the serotonin is the one that can enhance GIT contractions, because this serotonin is a vasoconstrictor. It is a vasoconstrictor. Then we see another cell as enterochromaffin-like cells, which we saw they produce histamine. And histamine is the one that enhances the action of acetylcholine and gastrin hormone. That is the work of histamine produced by the enterochromaffin-like cells. So these are the different cells we can talk about in the mucosa of the stomach and their secretions and their function. As we end this, there is what we call regulation or phases of gastric production. That is the last portion. We are going to talk about phases of gastric secretion, whereby these phases of gastric secretions include the cephalic phase. We have the cephalic phase, we have the gastric phase, and the intestinal phase, whereby gastric phase is purely nervous, it is due to nervous system, whereby this cephalic phase is just a thought. When you think about food, when you smell food, when you taste food, and when food reaches in the stomach, presence of food. Now this one is still thought. So thought, smell, taste, then sight of food triggers production of acetylcholine, and this acetylcholine, which is a neurotransmitter, is the one that stimulates production of gastric juice. So whenever you think about food, you see food passing around, you see it smelling, you start producing the gastric juice. That is the cephalic phase, purely nervous. Then the gastric phase, we talk about the presence of food type, presence of food bolus. When it reaches in the stomach, it causes distension or stretch. It causes the stomach to distend, and when it distends, it causes stretching. And this stretching, it is the one that triggers, it stimulates the stretch receptors and the chemoreceptors within the stomach. And these stretch receptors are the one that triggers what we call they trigger the Meissner's plexus to release gastric, to stimulate the parietal and the chief cells to release gastric. Because we see these Meissner's are going to release acetylcholine from the Meissner's plexus, or what we call the submucosa plexus, to release the gastric juice from the chief cells and the parietal cells. And that is the gastric phase, which is stimulated by presence of food in the mouth, in the stomach. This is when it has not even reached. You have just thought of food, but here food is already in the stomach. So the stretching of the stomach triggers the sensory receptors in the stretch and in the chemoreceptors, which causes the submucosa plexus to release acetylcholine, which causes or triggers the production of gastric juice. Then lastly, it is intestinal phase, whereby presence of food in the intestine, that is in the duodenum, presence of acidic food chyme in the intestine will trigger production of one gastrin hormone, another one production of secretin hormone. Secretin hormone inhibits production of gastric juice, whereas gastrin, in the case when food chyme has reached in the duodenum and it senses there is undigested fats of short chain, senses the polypeptide bonds were not broken, we see it producing more, triggering G cells to produce more gastrin hormone, which tells the stomach, please, you are not finishing your role. Can you complete it? That is intestinal phase due to presence of partially undigested. When the acidic food chyme reaches the duodenum and sees partially undigested proteins, partially undigested short chain fatty acid, it triggers G cells to release gastrin hormone, which causes further production of gastric juice. But if it finds food is well digested, then it produces secretin from S cells, which inhibits production of gastric juice. Thank you so much for listening. This is Medical Sciences Made Easy. Please always ensure to subscribe. If you haven't subscribed, subscription is once. Don't subscribe twice. It is once. But always like the video and share your comments for improvement. Thank you so much.